Hello all, welcome to Life Science Library. In this video we are going to see about few previous year CSIR NET questions from nervous system in unit 7 system physiology. In this video we will be covering previous year questions from 2020 to 2016. So all the questions from nervous system in this time period will be covered in this video. Hope this video will be helpful for CSIR NET and GATE Life Science students. First question from CSIR NET November 20 Session 2. Electrical response of afferent nerve terminal in Pacinian corpuscle after application of different grades of pressure are proposed in following statements. First, understanding the first statement. What is Pacinian corpuscle? Pacinian corpuscle is nothing but encapsulated ending of sensory nerve that act as a receptor for pressure and vibration. Consider this as a nerve ending of a sensory nerve. It is encapsulated. This is called as Pacinian corpuscle. It acts as a receptor for pressure. It acts as a receptor for vibration. This is all about Pacinian corpuscle associated with sensory function, sensory nerve fiber. Okay, we have understood the first statement. Second statement. Non-propagated depolarizing potential or the receptor potential is elucidated when small magnitude of pressure is applied to Pacinian corpuscle. First statement. Second statement. Magnitude of receptor potential is increased as the pressure to Pacinian corpuscle is increased. Third statement. Action potential is generated when receptor potential attains the critical value. So we are going to find the incorrect statement. Incorrect statement. Receptor potential shows all or none response. Receptor potential is not a graded potential. First we can use the elimination option. So we can find what is receptor potential. Receptor potential will not show all or none response. Receptor potential is a graded potential. So D is an incorrect statement. Receptor potential is a graded potential. It is not a graded potential. So it is also an incorrect statement. So option D and E is the best answer. A, B, C and D are correct statements. Non-propagated depolarizing potential is nothing but this is a wave. Depolarization, hyperpolarization and repolarization occurs. This is another wave. This is propagating wave. Non-propagated in the sense we will see only one curve. The wave is not propagated. This is the meaning of first option. Meaning of second option. Magnitude of receptor potential is increased as pressure is increased. This is simple logic. Action potential is generated when receptor potential attains critical value. There is something called as critical value, that is minus 55 MV, beyond which the threshold potential crosses this only, then action potential will be generated. So this option is very, very correct. So the incorrect statements are D and E. D and E is the right answer. Next, we are going to see this statement, this question. Which one of the following is not released by sympathetic preganglionic neurons? According to autonomic nervous system, we have two categories. That is sympathetic category, sympathetic neurons and parasympathetic neurons. Sympathetic neurons are help in their catabolic function. They increases heart rate. They are produced during emergency situation. They are fight and flight conditions. Whereas parasympathetic condition, they are help in digest digestion of food, and they are stimulated during rest. This is the basic understanding and overview of autonomic nervous system. First, analyzing all these. What are all these? These are neurotransmitters. Various kinds of neurotransmitters. We will understand this question that they have their own nature. For example, without all these things, analyzing each category. First up, neurotensin. Analyzing what is neurotensin. We have the first option as neurotensin. What is neurotensin actually? Neurotensin is a neurotransmitter. First point. They can also act as a modulator. This is the second point. Why? With whom they are acting as modulator? They are acting as modulator in cholinergic, dopaminergic, glutaminergic and GABA. They are acting as a modulator for these neurotransmitters. They are also involved in cytoprotection. What is cytoprotection? By modulating the mucous membrane. Cytoplasm protection, that is cytoprotection. Collectively called as cytoprotection, maybe not cytoplasm, they are collectively called as cytoprotection. These are the functions of neurotensin. Whom they produced? They are produced by hypothalamus and small intestine. First up. Second, we have serotonin. Serotonin. What is serotonin? Serotonin is also a neurotransmitter. They are sympathetic in function and they intervene with adipose tissue. They intervene with adipose tissue. They are also a neurotransmitter. Next, substance P. They have parasympathetic function. Main function of substance P is pain. P. So P. Pain perception. Substance P. Substance P. Pain perception. Substance P associated with pain perception. Also have a function with immune cells, cell proliferation, hemotaxis, and they are parasympathetic in function. Next, we are going to see about endorphin. So, what is endorphin? Endorphin is neurotransmitter. First point. They block pain signal to spinal cord. We have another interesting thing, that is enkephalin. So what is enkephalin? It is also a neurotransmitter. They block pain signal to spinal cord. Very sorry, I correct my mistake. Endorphin block pain signal in brain stem. Block pain, enkephalin block pain signal to spinal cord. Endorphin block pain signal to brain stem. So, long story short, I will summarize all the five neurotransmitters we have seen today. That is neurotensin, serotonin, endorphin, enkephalin, and substance P. Neurotensin, sympathetic in function, dopaminergic, cholinergic, GABA mediated, production in hypothalamus and small intestine. Serotonin, again a neurotransmitter, intervene with adipose tissue. Endorphin, they block signals to brain stem. Enkephalin, they block signals to spinal cord. Substance P, associated with pain perception. Another important thing we are going to see now is the substance P. Let me do it in black. Okay. In substance P alone, a parasympathetic in function. Sympathetic in function, not in production or release. Speaking about only function. Serotonin is sympathetic, but it is not released by neuron. It is produced by midline and hypothalamus. Okay, it is not released by neuron. It is also an sympathetic, but it is not released by neuron. It has parasympathetic function. In otherwise cases of neurotensin, endorphin, enkephalin, all are sympathetic in function. Serotonin is not released by neuron, and substance P is parasympathetic in function. Now coming back to this 14th question. Which one of the following is not released by sympathetic preganglionic neuron? We can very well say that these have neurotensin have sympathetic function, substance P have parasympathetic function, enkephalin have sympathetic function, serotonin have sympathetic function, but it is said that which is not released by sympathetic preganglionic neuron, that is serotonin is not synthesized by sympathetic preganglionic neuron because they are produced by hypothalamus midline. Hope so. This 14th question from Csnet November 2020 session 2 is clear. Next question. Mammals' primary circadian clock is located in which of the following parts of brain? Suprachiasmatic nucleus in pineal gland. We know about occipital, this is school level studies. Frontal lobe is also known to us. Amygdala is not an option. So suprachiasmatic nucleus present in pineal gland act as circadian clock of human. So, next question from CSIR-NET 2019. Fatafat. In nervous system, action potential is generated at axon hillock. In physiological condition, it is conducted to the terminal end of axon. The location-specific origin of action potential to its generation-specific conduction are explained by a researcher in following proposed statement. The membrane of axon, we are going to find correct statement. Membrane of axon hillock has highest threshold for generation of action potential. No. Membrane of axon hillock have lowest threshold of action potential generation. So, A is wrong. Membrane of axon hillock has large number of voltage gated Na plus channel and it make it more excitable. Correct statement. So we have arrived at answer, but we will check out other statement. Propagating action potential in the middle of axon cannot generate another action potential in the direction of cell body since large fraction of voltage gated Na plus channel in the preceding portion of the voltage is inactivated. Correct statement. As the number of voltage gated Na plus channel is less in the preceding portion of the axonal membrane, the propagation of action potential in the middle of axon cannot generate another action potential in the direction of cell body. This is wrong statement. So B is the correct answer. B and C are the correct statements. Next, CSIR-NET December 2018. Conduction velocity of action potential in the myelinated nerve fiber was much greater than that of an unmyelinated fiber of the same diameter. Which of the following proposed statements explain the observation? Speed of conduction of nerve fiber is determined by plasma membrane resistance and axial resistance of axonal cytoplasm. We are going to find incorrect statement. Myelin sheath decreases effective membrane resistance. So we are going to find what are the correct statements. B is the correct statement because electrical property of myelinated and non-myelinated fibers are not similar. So B cannot be an answer. Speed of conduction of nerve fiber is determined by plasma membrane resistance and axial resistance of axonal cytoplasm. This is a wrong statement. And now, myelin sheath decreases the effective membrane resistance. This is a wrong statement. Myelin sheath has nothing to do with effective membrane resistance. Magnitude of an electronic potential decreases with more distance. Magnitude of electronic potential decreases with more distance along axon in myelinated fiber than on non-myelinated fiber. This is also an incorrect statement. C, D is correct answers. Electrical properties of myelinated and non-myelinated fibers are not similar. This is very correct statement. So A and B cannot be an answer. Voltage gated Na plus channel are highly concentrated in the nodes of Ranvier. This is correct statement. So E can't be an answer. Arriving at an answer, answer C is the right option for the CSIR-NET December 2018 question. Now, look at this question. Mammillary bodies. This is a straightforward question. Mammillary bodies are present in I give a clue. Mammillary bodies are present in diencephalon. Diencephalon. Find which it is diencephalon. Whether midbrain is called diencephalon, epithalamus is called diencephalon. Diencephalon includes so many components in brain. All these components, the components of diencephalon. The right answer is, hope you found the answer. Mammillary bodies are present in hypothalamus. Hypothalamus is the right answer. This is a straightforward question. Now, this question needs more explanation regarding Group 1B sensory fibers, Group 1B sensory nerve fibers, Group 1A sensory nerve fibers, Group 1A sensory nerve fibers. I will be dealing with a separate video on nerve fibers, on sensory nerve fibers in detail. As of now, we will know how to arrive answer to this question. Next video will be on nerve fibers, sensory nerve fibers, and fiber types A, B, C. When skeletal muscle was passively stretched, it is contracted reflexively. However, when the muscle was overstretched, in this way it showed sudden cessation of contraction followed by relaxation. Following statement provided the possible explanation of this observation. Passive stretching of muscle causes stretching of muscle spindle. This is a correct statement. So what is passive stretching? We know what is this is something that is occurring called as passive stretching. Then what is active stretching? Active stretching in the sense you do something to stretch. This is active stretching. Passive stretching, you never do anything but stretching occurs. Just like a recoil, it is called as passive stretching. This is a correct statement. Overstretching of muscle stimulated Golgi tendon organ. So what is Golgi tendon organ? Consider this as a muscle and consider this as a bone. Muscle get attached with the bone. Here we have a Golgi tendon organ. If this is stretched to a large extent, this Golgi tendon organ will have a greater role for it. So A and B are the correct statement. A is the correct answer for this question. Next question from June 2017. Following are some statements about the mechanism of stimulation of receptor for touch, pain, vision and warmth. So we know light. We have seen this in GPCR signaling, phototransduction, phototransducin, arrestin, alpha, GTPase, GDP, GTP switch, C cyclic ANP, adenylyl cyclase. We have seen this in this GPCR signaling. Hope this question becomes very easy if you have good knowledge in cell biology and cell signaling. Without reading this, we can see that light causes closing of Na plus channels in the outer segments of rods and cones. This is a correct statement. We have arrived with an answer, but still we will check out other options. Pain sensation is caused by opening of Na plus or Na plus Ca2 plus channels in free sensory nerve ending. This is a new learning. So, pain perception is done by substance P. What happens if you have a pain? Pain leads to opening of Na plus channel. Na plus channel will open. Or calcium 2 plus Na plus channel open. This will happen during pain. Only because of this we are sensing pain. Touch receptor does not require any voltage gated cationic channel for active. This is a wrong statement. So A is a wrong statement. B and C are the correct statement. B is the right answer. Again from CSIRnet June 2016. Membrane potential in gain squid axon recorded intracellularly at resting condition minus 70 MV, that is resting membrane potential, was reversed at a peak action potential of plus 35 MV. After stimulation of nerve fiber with a threshold electrical stimulus, this overshoot of membrane potential has been explained in the following proposed statements. Okay, we will read all the statements. We are going to find the correct answer. Rapid increase in the Na plus conductance during the early phase of action potential causes membrane potential to move toward equilibrium potential of Na plus 45 MV. Okay. Next, Na plus conductance quickly decreases towards the resting level after the peak in the early phase, and Na plus are not able to attain its equilibrium potential within short period of time. Okay. Conductance of Na plus in early phase of action potential. This is okay. There will be any conduction in Na plus in early phase of action potential? This is a bilkul galat. This is an incorrect statement. So these two options are neglected from the game, not in game. So remaining option 4 is neglected, but still we will read option 4. Increase in K plus conductance due to stimulation of nerve occur before the changes of Na plus conductance is initiated and thus causes overshoot of peak of no. Overshoot will occur only because of Na plus, that is depolarization. So fourth option is also wrong. You will read about second thing. Na plus conductance quickly decreases towards resting level after peak in the early phase, and Na plus ions are not able to attain the equilibrium potential within short period of time. First statement and second statement are correct statement. They will not able to attain the equilibrium potential within a short period of time because 3 Na plus ions move out and 2 K plus ions comes in. This is the natural law. B option is right. Next question. Cell bodies of sympathetic preganglionic neurons are located in. This is a straightforward part B question. Intermediolateral cell column of spinal cord, posterior cell column of spinal cord, celiac ganglion, paravertebral ganglion. And what is the correct statement? Cell bodies of sympathetic preganglionic neurons are located in intermediolateral cell column of spinal cord, that is gray matter. Gray matter of spinal cord. Next question from Csirnet June 2016. So we will have 3 questions left for the session to end. Okay. From Csirnet June 2016. Oh, we have read the statement. Already the gain squid question. Okay. Moving to the next question. Which one of the following does not occur due to the stimulation of baro? This is a homework question. We have a homework for our viewers. That is, what is baroreceptor? Baroreceptor will occur, will function when there is any change in the pressure. This is barometer, something related with baro is related with pressure. So, baroreceptors will occur, will stimulate only if there is a change in the pressure. Will bradycardia will not occur due to stimulation of blood pressure when it associated with change in the pressure. The answer is vasoconstriction, because vasoconstriction, I am not okay with giving explanation to this question. Viewers, please take this as a homework and comment in the comment box below, as our usual method of explaining all the 4 options clear cut. Fatafat, pause this video and give explanations and suggest whether D is right or wrong, B is right or wrong, C is right or wrong, A is right or wrong. Viewers, quickly we will see about December 2016 this question. External pressure given on a mixed nerve causes loss of touch sensation while pain sensation remains relatively intact. Okay. On the other hand, this is a learning. That is external, when pressure is given, when lot of pressure is given, loss of touch will occur. This is a learning. On the other hand, application of local anesthetics on the same nerve induces a loss of pain sensation, keeping touch sensation least affected. This observation can be clearly explained by the following statement. We are going to find the incorrect statements. External pressure causes loss of conduction of impulses in small diameter sensory nerve fibers. So, external pressure causes loss of conduction of impulses. This is illogical actually. So, A is incorrect. Local anesthetic depresses the conduction of impulses. No, this is incorrect. A and B are the 2 incorrect statements. We will see about what is fiber A, what is fiber C when I explain about the chart that I saw that we saw in the previous questions regarding sensory fibers. Fatafat, next question from CSIR Net, June 2017. Action potential, regarding action potential and aftermath of hyperpolarization. So we should know what is hyperpolarization, what is depolarization, what is repolarization. We should know about all these things. First of all, consider this as a graph. At resting membrane potential, that is minus 70 MV. At 55 MV, we reach the threshold potential. That is, we will reach the peak when depolarization. This is repolarization, and this is minus 90 MV. This is hyperpolarization. Back to our baseline. This is what about an action potential generation. This is the basic detail about nervous system. Next, about this question. Action potential of nerve fiber is described by different components, including aftermath of hyperpolarization. Mechanism of generation of this after hyperpolarization has been proposed in the following statement. Increase the conduction of Na plus has returned to the basal level, but the conduction of K plus remains elevated during after hyperpolarization phase. Fatafat, think an answer with a graph that I explained just now. Students, are you here? Please think an answer. Next option. Membrane potential is pulled even closer to the K plus equilibrium potential after hyperpolarization phase, or conduction of Na plus is increased before any change in K plus conduction during after hyperpolarization phase. So during after hyperpolarization, what is happening? At after hyperpolarization phase, the membrane potential is driven close to the Na plus equilibrium potential. A is correct statement? Correct. Increased conduction of Na plus has returned to the baseline level, but the conduction of K plus remain elevated during after hyperpolarization phase. So A is the correct statement. Other things are wrong. We are going to find the correct statement. We will see whether B is right or D is right. After hyperpolarization phase, membrane potential is driven close to the Na plus equilibrium potential. So we say it's very hard to bring Na plus equilibrium. What will be the right answer? Membrane potential is pulled even close to K plus equilibrium potential after hyperpolarization. What can be the possible answer? I gave all the explanations. Hope you can now guess the answer. As option A is right, because it is very hard to bring Na plus to equilibrium. So option A is the right answer. Hope with the graph I explained, you can arrive at answer A. Close to K plus equilibrium potential. Increased conduction of Na plus has returned to the baseline, but the conduction of K plus remain elevated. The membrane potential is pulled even close to the K plus equilibrium potential after hyperpolarization phase. So A and B are the right answers. Okay. Okay then. We have seen all the questions so far from CSIR Net. Life science examination is concerned with system physiology without except this question, because in this question with the previous year question bank that I have, I don't find the graph. Without that graph, this question this question is hard to be explained. So then we have seen all the questions from 2016, 2020. All the questions with the basic concepts are explained in this video. Do refer this video with all your friends and fellow aspirants, and I hope This video is very helpful for CSIR NET Life Science students and GATE Life Science students. Thank you for watching this video. Please do like, share, subscribe to my channel. Thank you.